Nobel laureates, excellencies, ladies and gentlemen. It's a great pleasure and privilege for me to welcome you all to Karolinska Institut's Aula Medica for the 2017 Nobel Lectures in Physiology or Medicine. My name is Ole Petter Ottersen. I'm Director Vice Chancellor of Karolinska Institutet. During the Nobel Week, and particularly during the Nobel Lectures, we celebrate science and rejoice at the unique contributions of some truly exceptional individuals. Because research is a way of quenching our thirst for knowledge, testing our own limits, and having an outlet for our creativity and competitive spirit. Within physiology or medicine, the ultimate goal is to achieve scientific breakthroughs that change our views of human health, disease, and normal vital processes. Dear laureates, you have surely contributed to this. Today, it's an honor for me to extend my warmest welcome on behalf of Karolinska Institutet to Jeff Hall, Michael Rosbach, and Michael Young, the 2017 Nobel laureates in physiology or medicine. They are receiving the Nobel Prize for their discoveries of molecular mechanisms controlling the circadian rhythm. Without further ado, I now call upon Yulin Sirat, Professor of Clinical Integrative Physiology and member of the Nobel Committee for Physiology or Medicine. She will introduce the laureates. Yulin, the floor is yours.
It is a great honor for me to introduce the 2017 Nobel laureates in physiology or medicine, Jeffrey Hall, Michael Rosbach, and Michael Young. They have received this year's Nobel Prize in physiology or medicine for their discoveries of molecular mechanisms controlling the circadian rhythm. Life on Earth is adapted to the rotation of our planet. For many years, we have known that living organisms, including humans, have an internal biological clock that helps them anticipate and adapt to the regular rhythm of the day. But how does this clock actually work? Jeff Hall, Michael Rosbach, and Michael Young were able to peek inside our biological clock prizes along their distinguished careers, including the Gruber Prize in Neuroscience, the Louisa Gross Horwitz Prize, the Shaw Prize, the Wiley Prize in Biomedical Sciences, and the Gardner Foundation International Award. As they say in the field of chronobiology, it is time to hear from each of the laureates. And I'm the timekeeper for this afternoon's session. We'll hear 3 individual lectures, and these journeys have likely been fueled by scientific inquiry, little mischief, hard work, teamwork, maybe a little friendly competition, and of course, one cannot forget their love of genetics and that of the fruit fly. So now I'll begin with the introduction of the first of our 3 laureates, and this is Professor Emeritus Jeffrey Hall, chronobiology. Professor Hall has spent his career examining the neurological components of fly courtship and behavioral rhythms. Now I'd like to ask you all to join me in welcoming this year's Nobel laureate, Professor Jeff Hall, up to the stage, and we will hear a lecture from him entitled The Little Flies, Multifaceted Basic Research Coming Out Better Than Intended. Professor Hall.
Do I just start talking?
You just start talking.
Okay. And further words of gratitude to certain specific biologists and geneticists. So, for example, not too long after I was attempting to establish type of chromosomally based variant. Now, back to the matter of another type of chromosomal variant that I've already referred to that includes the designators yellow and forked. It was Muller himself who generated that now classic arcs. Another corner of the overall biogenetic enterprise, in principle, a priori, might involve Drosophila behavior as opposed to developments, starting with the fertilized egg. Most conspicuously postnatal behavior after you get the mature adult animal. And so behavior biogenetics was pioneered principally, conspicuously, by the aforementioned Seymour Benzer, who by then had moved in physiology or medicine, oddly named, that there should be 5 awardees. There should be 5 awardees this year in this category. And one of them should be the little flies, which have contributed. You can't do this, of course. And it also, there can't be 5 awardees that flouts the rules, or as Americans would say, flaunts the rules. Most of them being illiterate. But awardee number 4 should be the little flies, because as exemplified here on the third rock from the sun. But to study for mutants with abnormal rhythms in fruit flies and then Neurospora and later a variety of other organisms, that those mutations had interesting abnormalities, but the mutations causing the mutant rhythm anomalies, the mutations also pointed to genes, or what in terms of American research grant jargon are PIs, the 3 PIs. That stands for principal investigator. Perhaps the adjective is correct, although and those two still do, are AIs, actual investigators. And I can't help give a tip of the hat to a subset of those who voted with their feet and their pocketbooks to travel long distances, because I realized that there were all these fine AIs nominally under me, but also in Michael's lab. People, I know that some of them have made this trip. If you just keep going, even if for a while, if nobody's necessarily paying any attention, but it should have been. But the same happened, again, better than we intended. It'll be funky, but warranted. So I'm done. Well, I'm really done in life as well as, but if there's been any attention, I thank you for it.
Business since the 2nd of October at 5:10 in the morning Boston time has left me virtually speechless. But being speechless is not really appropriate here, is it? So I'm afraid I'm going to have to screw up my courage and try and say something. And so a simple systems view of circadian clocks, a so-called skinogram named after Arnold Eskin, one of the early pioneers in the field, is illustrated here. And it simply demonstrates the fact that there are input signals, principally light, also temperature, which entrain the clock, are not necessarily the ones which have staying power. And so at Brandeis, Jeff and my lab, who associate with, co-mentor, if you will, in the case of John, Patrick Emery in my lab, who's not here, collaborated with Ralph on the cloning of cryptochrome. So, and then of course, this had an element, a strong element of personal friendship. We had, thank you very much.
After a summer of research with Burke Judd, and probably this emphasizes the importance of summer internships, he was working with the Drosophila genome, and that probably spurred on his interest in science because he remained at the University of Texas to complete a PhD in genetics in 1975. Michael Young continued his studies throughout his postdoctoral training at Stanford University School of his Nobel lecture entitled Time Travels, a 40-year journey from Drosophilus clock mutants to human circadian disorders.
So as Michael pointed out, there's a great deal of homology when we move from the fly to other animals, the rest of the animal kingdom. So in us, for example, we have many familiar proteins. We have clock and BMAL, which are the orthologs of clock and cycle in the fly. We have, in collaboration with Steve Kay in 1997, and they had generated a form of PER which was connected to luciferase so that you could use a light monitoring scheme to image live Drosophila and all the intact and dissected body parts of the fly, and realized that there were PER-driven. Love to have been able to be here. Alina Patke, again, has been responsible for every aspect of the human work. She's had her hands on every part of that project, and she is here, and I just want to thank her personally. So thanks for your attention, and for the honor.
I would like to extend our gratitude to the Nobel laureates for impressive presentations this afternoon. May I ask the laureates onto the stage for a round of applause.